Good morning, everybody, and welcome to the Capsicum Pan-Genome Consortium webinar. My name is Yoni Rotter, EMEA and APAC Territory Manager at Energene. In the webinar, we will go over why is pan-genome needed, the implication it has in capsicum breeding, and the current status of the consortium. Joining me are Dr. Ilan Paran, a researcher in Israel Agricultural Research Organization Volcani Center, focusing on pepper genetics and diversity since 1993. Dr. Alan Van Dynes is the director of SITS Biotechnology Center and associate director of the Plant Breeding Center at the University of California, Davis. And Dr. Sharon Rechav. Sharon has been working in the ag biotech industry for over 8 years and with Energene for almost 3 years as a technical expert. We will dedicate the end of the session to live discussion, and we encourage you to participate and ask your question. To do that, you can use the raise your hand option to ask for permission to speak or simply write a question. Ilan, the stage is yours.
Okay, hello everybody. We know that capsicum pepper belongs to the Solanaceae family. There are several genomes that are available, but pepper has a complexity in two aspects. First is the very large genome, the largest than all other sequenced genomes. And it also is very rich in repetitive DNA. What are the available genomes to date for pepper? In 2014, two genomes were, well, more than two genomes, several genomes were published. CM334, which is a Chile type landrace by the Korean group of Kim et al. Later in 2017, a later and more improved version has been published. Another is Zunla by the Chinese group in 2014. We have a wild relative of Cyanum, which is Cyanum var glaburiosculum, again in 2014. And then more recently, a hybrid of CM234 using the 10x technology by the Alan Van Dynes group. These are all Cyanum, and in addition, we have one accession of C. chinense, which is a landrace, again published by in the paper of 2017, and Capsicum baccatum, a landrace which is resistant to anthracnose. Now, within the current pan-genome project, Maor, which is publicly available, a block type has been sequenced, and the results will be shown later in this presentation. I should mention also resequencing of close to 400 accessions, and which were analyzed for present and absence analysis, which was published last year. And also there are several ongoing unpublished projects in different countries, such as the Netherlands, South Korea, and China. This is a slide taken from Alan Van Dynes' publication in 2013, showing the diversity of different cyanum types. Which by using gene chip technology. Here we see the CM334, but we already see that there's much more diversity also in the many different Chile types, and also in the green non-pungent type. So CM34 represents just a fraction of the diversity within cyanum. What is missing? First, sweet types, mostly modern breeding lines of different types, such as blocky, long sweet, mini sweet, lamuyo, or cherry, and other types. Additional Capsicum annum chili types, such as, again, modern breeding lines, such as jalapeno, cayenne, New Mexican, poblano, as well as other types. Also included habanero, which is a seed chinense, but is a important type. And there's also very small representation of wild and exotic germplasm. What is the potential application of the pepper pan genome analysis? There are several high-quality Deneuve references. Genomes will assist us in detection of structural and SNP variation within the breeding material. This information will be useful for market development. It will also assist in analysis of gene content in segments of the genome that are not represented in the reference genome, and detection of wild or exotic introgressions in these breeding lines. I think I am finishing my part, and we will move to Sharon, which will talk about more technical aspects of the consortium.
Okay, thank you, Ilan. So I'm going to start with the bottom line that I think many of you may find interesting. So what do you get? Let's say you've joined the consortium, what do you actually get? So you get scaffold level de novo max assembly of your sample. It would be arranged into pseudomolecules, chromosome level. All of the different assemblies would be mapped to one another, creating a pan reference. By the way, all of these terms are going to be explained at length in just a minute. We are going to provide homology-based annotation of each one of them, and then we are going to use that annotation to compare them to one another for gene variations. And all the results can be explored in a few genome browsers, but preferably the freely available customized IGV that Energene has made public around a year ago. So now let's just take a step back and ask the question, why do we need pangenomes? Pepper has, as Ilan showed, a few reference sequences. Why not just use one of them to capture your diversity? So I've made a small illustration. The first reason is some variations between the two. If you're comparing resec data of a certain line, some variations would result in false discovery of SNPs due to copy number or structural variations. Furthermore, a lot or some of the reads may not align anywhere, and that's just diversity that you are not looking at because of the distance between your line of interest and the reference. Another limitation is the fact that this method can only detect small range polymorphisms like SNPs and small indels. If there are translocation events or other structural dissimilarities, you would not be able to detect them. So I'm going to show you just a few examples, just two, of the extent of these limitations. One is taken from tomato. This is a slide courtesy of Dr. Ruth Wagner, and it shows the percent of mapped reads of resequencing data from different accessions and cultivars of tomato to a single reference. As you can see, on average, 40% of the data is lost. You do not use it. Another example showing the second type of errors is looking at Maize data. This is actually a Maize assembly that we've done a few years back. And if you take the reads from that assembly and you align them to the publicly available reference of Maize B73, and you then compare the location of the SNPs to the actual locations on the Deneuve assembly, you find that 45% of them are not collinear, meaning that the order of SNPs of 45% of them not as the reference tells you. So these are two examples of why we need pangenome, and now I'll just present to you at greater length what we actually offer. Our PanMagic solution begins with selecting key lines. That's obviously a choice of the consortium participants. It should cover the diversity that Ilan has just shown at best, and also capture the interest of the consortium members. After those key lines are selected, DNA is extracted, sequencing data is produced, and we assemble these lines, arrange them into pseudomolecules so that we can go on to the next phase. Another comment here is, since pepper is an inbred and we want to reduce the errors as much as possible, have a comprehensive analysis, we encourage the lines to be as homozygote as possible. That's something to consider. Now, after we have that, we continue and provide an all-to-all genome mapping that I'll explain more about. And that basically gives you an infrastructure that is not biased to a single reference, which you can use for several analyses. We provide one of them that is based on genes, and I'll explain more about it as I go on. De novo assembly. So what we currently have after a few years and a few hundreds of assemblies behind us is the De Novo Max. It's a cost-effective yet very high-quality De Novo assembly. The data is a combination of 3 different sequencing methods. One is paired end sequencing, 250 base per read length. The next is mate pair with 2 to 4 kb insert size. And the third is 10x chromium. That's very efficient and cost-effective synthetic long reads. Overall, the coverage of the genome is 95x, and these different data types are used in combination to produce the assembly. Now, these are examples of the NovoMax assembly results. You can see here a bunch of species. We've already tried that on, and I'm going to concentrate on the Mao variety, which we've just did for Ilan as part of this consortium. It's a good length, 3.1 gigabase. It's extremely, its contiguity is very high. You capture half of the assembly length in only 9 scaffolds, the smallest of which is 140 megabases long. We basically captured 3 full chromosomes within that. Also, the N90 is very good. The Busco percentage, meaning how well do we capture genes, is also very high. So this is the assembly level that we can reach. Moving on, after you have a few de novo assemblies, these are schematics that represent 6 different de novo assemblies of 6 different varieties. I'm going to show to you what we actually do, comparing these 2, purple and green. So we start with linear genome mapping. This is basically, we walk along all chromosomes and we depict at base pair resolution all the places that are matched, meaning there's a full sequence identity between the 2 lines, and all the regions that are unmatched are stored also. In this way, if you are sitting in a certain position, let's say here, this is a a region that is absent from this purple region, found only in this green one, you don't know the exact location because there is no, but you can map all the sequences here and say they should be around this position between the two adjacent matching regions of the other. After we have that, we continue on to homology-based annotation. We take an existing transcript database. It can come from ab initio annotation of these genomes or just pulling from a public source. We take all of them and apply homology-based annotation on these two genomes. After we do that, we use the linear mapping to give them colors. These are actually colors in the output file that you can view in a browser. And the colors tell you the type of variation that you have. For example, blue signifies a match. A match means that these two genes have the same copy number and are located approximately in the same position in these two genomes. A light green color, like you see here, basically says this gene is present only when compared to the purple assembly. This gene appears only in this green assembly and not in the other. And minor translocations have a light khaki color. Major translocations, perhaps to an entirely different chromosome, have a darker khaki color. Extra copies are marked in this pink color. We do all this analysis. The linear mapping is done all by all. So if, for example, you have 6 genomes in your pangenome, you get 15 such coordinate conversion files. And the annotation is done comparing all of the genomes to the chosen pivot of that project. This is how it actually looks like in IGV, now customized IGV. This is taken from Maize. So at the upper part, you see an annotation tracks, exons and introns. I zoomed in into 2 genes of the reference that you are now using. And then just underneath, you see 4 different tracks that signify the comparison to 4 different de novo assemblies. So this gene at the right, when compared to these 4 different assemblies, 3 of them are a match, and 1 of them is absent. It has no copy of that gene. Looking at the left side, 2 of them are absent and 2 of them are translocated. If I'll now take this bottom genome and I'll reverse, and I'll now use it as a reference and I'll compare it to this line that was the reference before, what you get is you actually only see the annotation of this gene and a match, and at the left you see nothing because this gene is actually not there. So this is how it looks like. What can you also do with these pangenomes? So in the short term, you have a cost-effective multi-reference genome that you can use. You can map sequences, cast it, and each result that you get can be mapped to any of the other ones because they have this conversion method. Obviously, you can access the full sequence of all the assemblies since each participant contributes 1 and gets access to all. You get the PAV-CNV results in a readily visualized output that you can explore, and you can determine allele sequence of the different varieties, and as I said before, you can place sequences on top of it. That's the short term. What can you do in the future is, for example, you can use that as the basis for a new genotyping assay, an array or an amplicon set that you can use in your day-to-day. You can use it as infrastructure for sequencing-based genotyping capability. That's actually something that we at Energene regularly do. You can impute your legacy data, meaning you can take existing SNP data and map it on top of it and then when you have a match, you can impute to full sequence, if possible. You can use it in your day-to-day to facilitate the development of new trait markers, and you can use it as the starting point for a haplotype database and allele discovery pipeline. Either you can do it with us or you can do it yourself. And at this, I want to return the floor to Yoni to present the actual commercial offer.
Okay, thank you, Sharon. So to summarize, the consortium is led by Dr. Ilan Paran and Dr. Allen van Dijns. The idea of the consortium is to de novo assemble to reference level several genomes, organize them to pseudo-chromosomes, and perform all-to-all chromosome mapping to capture the broad diversity that exists in pan-genome architecture. Actually, by joining the consortium, you pay for your own line while receiving the full assemblies of all the participating genome, including a pan-genome analysis that Sharon just mentioned. To join the consortium, each participant contribute an accession of their choice. The cost for joining is $59,000. The deadline to join to the consortium is December 2019. During PAG 2020, I would like to invite you to invite the consortium members for a face-to-face meeting to provide all the updates and to have decisions that need to be decided. At the moment, we have 4 new genomes from the academia and 3 different commercial companies. We have 1 genome from Volcani Center, which we already delivered outstanding results. We have Top Seeds International. We have People Seed company of Dr. Jonathan Elkind. And we just got also Nunems as part of BSF group. As you can see, we had a great start, and we welcome you all to contribute your genome to the consortium as well. Now I will open the floor to Q&A, and I see we have some questions already. Daphne, how can I read this question? Alan, Ilan, and Sharon would be happy to take your question now, so please feel free to write anything, or we will open your microphone. Okay, one's asking about budget. If we don't have the budget found to contribute for a single genome, would it be possible to work together with other researchers? Yeah, so of course, if you don't have, if you don't have the budget for a consortium to join to the consortium as a member, there is a way to get your name, to get your email address, and we will try together to join 2 researchers together in order to share the budget, and you will participate actually together to get the access to all the others. Do we have another question? Okay, what if I don't join on the specific deadline? Is it possible to join later? Okay. So in principle, the idea of giving a deadline is to deliver the pan-genome analysis as fast as possible. Each member that join to the consortium will get separately his genome very, very fast. For example, the Mao variety we did almost in a month. We guarantee to deliver a genome in 4 months, but actually we do it in few. But the idea is not to wait with the pan-genome analysis, and this is the reason we want to finalize the list and to start with the pan-genome analysis. But if you will be late, there is an option to join later, but it will usually it will cost more, but we hope not to get to this option. Any other question? Alan, do you want to add some comments?
Yeah, my only comment was, you know, this maybe it's more technical. If you put up the second last slide, you have 3 genomes available right now, and there's the one public genome. And you had a question. It's important to have a publicly available reference, and we've thought about this a lot. There's, you know, as Dr. Paran mentioned, there's several genomes available out there, but I would contest that the Mayor genome, the Intergene just finished, and also our 10x genome is perhaps the only one that is of good enough quality, as far as, yeah, of good enough quality. The previous genomes really don't have good quality, especially in the repeated regions that the 10x genomes, like Mayor and CM334 hybrid that we did. So I would suggest that, you know, the publicly available reference be the CM334 hybrid in my case, you know, or Mayor. And it's really, we published this in our publication on the 10x genome that, you know, yeah, the contiguity or the order of contigs is really not very good with the other genomes.
Absolutely, it's which reference will be chosen to be the reference one for the pangenome analysis is up to you and Ilan, and we will make the internal discussion, and we will make the decision.
Yeah.
Okay. We have another question from Ronit. Hi, Manishma. What is the schedule for sequencing and pan analysis? So usually the assembly, well, sequencing usually takes, it's not up to us, it's to the sequencing center, it usually takes up to a month. It can be even more, but usually that's what it takes. The assembly itself, we guarantee to deliver it within 4 months, but we usually do it much faster. And the pan analysis would need to wait until all assemblies are done. And once they are done and the consortium is closed, we've finished assembling all of the lines, it should take us a few months, 3 to 4 months to complete.
This is Alan. This is it's a question I know the answer to, but I think it's important for people to understand. We talked about this. For this to be successful, given the technology, it's important that people contribute homozygous lines, or as homozygous as possible. So hybrids really are not going to work well here. And then correct me, I think you want leaf tissue. If you can comment on, you know, what type of materials you want, that'd be great, Yoni.
Okay. So regarding the tissues that we have a standard how to collect tissues, plant tissues, and I can send to each of one of you that interested to get exactly how you should grow the plant. We have a very strict and clear instructions.
Protocol. Maybe Ilan can attest because he's just recently done it. If there are any special requirements, or is it difficult or hard to provide?
Well, basically use the young leaves. You have to make sure you use young material, and before DNA extraction, you put the plants in dark for 48 hours. basically then collected the leaves, and we send the leaf material to the company in the US, which they are specialized in extracting high molecular weight DNA, and they send it directly to the sequencing center.
Okay. Do we have any more questions? Okay, so thank you very much. I would like to thank Ilan and Alan and Sharon for the time today and the terrific presentation. Thank you very much. The recording of the presentation will be sent to all the participants. You will have it available in the next few days, so keep an eye out, and feel free to contact us through the website or directly to my email address. Thank you for joining today.